Okay, so in the final part of this session, we're going to look at the role of antibodies and antigens in testing for SARS-CoV-2. So before we get on to how these tests work, we need to look at antibodies and look at what antibodies are. So antibodies are these proteins that are secreted by cells of the immune system to recognize anything that is foreign or not host. Antibodies are created by our B cells. They're a type of white blood cell, and these B cells, as they are developing, they are checked to find out whether or not they produce an antibody that recognizes ourselves, as in the host. And if that B cell produces an antibody that recognizes the host, then that B cell is destroyed. And what effectively happens with B cells is they produce effectively random antibodies. And these random antibodies could recognize any potential pathogen. So they're not designed to recognize a particular pathogen. We just create millions and millions of them and hope that one of our B cells creates an antibody that recognizes the pathogen, and that antibody then comes in contact with the pathogen and helps to neutralize it. So there are two ends of an antibody. And the antibody is made up of 4 protein chains, so 2 light chains and then 2 heavy chains. And there are 2 ends to the antibody, the variable region, and this is the bit that differs between each and every antibody, and the constant region, the FC portion, which is the same between each antibody. So this variable region, the B cell makes, each B cell makes a different antibody with it has a different DNA sequence coding for a different protein sequence for this variable region. So each individual antibody has the potential to recognize something completely unique. So the way antibodies work is this variable region binds to the pathogen. The antigen is just something that the antibody binds to. That would typically be a single protein from a pathogen. So the antibody binds to it, and then the FC portion is sticking outwards. And if an FC portion is sticking outwards from an object, a bacterium, a virus, a protein, something that the antibody is bound to something effectively very small but solid, then other immune cells such as macrophages will recognize that this antibody is bound to a relatively large immobile object, such as a bacterium or a virus, and then the macrophage will come and engulf the whole thing. Now this is where the immune system gets complicated, and you'll learn about this at some time in the future, but that macrophage that has engulfed this antibody and virus can then start to chop up that virus and effectively present bits of that virus on its cell surface to other B cells, and trying to find a B cell that can recognize something that the macrophage has presented on its surface. So if the macrophage has engulfed the virus, chopped up those proteins and presented some of it on its own surface, and then this binds to either a B cell or a T cell, a different immune cell, that can activate those B cells and T cells and make them expand, and we call that clonal expansion. And then those really proliferate, and we get large numbers of cells that recognize that viral protein. So if a macrophage has engulfed a virus, chopped up those proteins, presented them on the cell surface, and then presented them to a B cell, and that B cell recognizes that peptide on the surface of a macrophage, then that B cell will become activated to proliferate and produce vast amounts of antibody that recognizes that specific peptide. Now that might be quite complicated, and that's a brief summary of your second year immunology, so do not worry about it too much at this stage, but that is roughly, with a few steps skipped, that is roughly what happens. So we're talking about antibodies. Antibodies are part of the acquired immune system. B cells create random antibodies that could recognize a pathogen. If that B cell comes into contact with that pathogen or a part of that pathogen presented on the surface of another cell, that B cell will become activated, it will proliferate and produce vast amounts of antibody to then mop up any other of that virus that is lurking around. Those antibodies that are secreted are proteins which bind to the specific target, and we talk about an antibody target as being an antigen. That is just part of in this case, virus, but it could be part of a bacterium. So antigens and antibodies, what we're now going to talk about, we'll talk about the antibody is the thing that recognises the antigen. So we can look at antibodies from the point of view of what happens during either infection or immunisation. So if we are infected with virus A that contains antigen A, that could just be a surface protein, what we find is a couple of weeks after exposure, antibody levels rise. So this is what we call the primary anti-A response. That means that we've got blood serum antibodies to virus or antigen A. Levels go up, then levels go down again. If we then re-inoculate with that same antigen, you get a much stronger secondary response. And this is where the B cells have initially been primed to produce the antibody, and then they've been primed again to proliferate, and this is more of a long-term memory response. So if you encounter something twice, you get a much more potent response. Here we're encountering antigen B. At this point we get the same strength response as the A response the first time round. So this is the whole basis of immunisation. We often do immunisations with 2 or 3 sequential vaccinations where we inject a protein from the pathogen once and then a few weeks later inject it again and maybe even inject it a third time. And each time we see an increase in the amount of antibodies that are produced. So these antibodies are really useful tools because they very accurately bind to a single specific target, which means that we can use them in clever ways to detect viral proteins in experimental systems or in diagnostic tests. And we can create antibodies experimentally, usually by inoculating an experimental animal with a specific protein from, let's say, a virus. So we can take a rabbit, inject it with antigen A, and the rabbit will produce antibodies to antigen A, and then we can extract those antibodies from the rabbit's blood and then isolate them and use them in our diagnostic tests. So one of the first things that was done after SARS-CoV-2 was identified was to generate antibodies to it by injecting experimental animals with either the virus or just individual proteins, and generate lots of antibodies that specifically recognise maybe the SARS-CoV-2 spike protein, for example. And what we can do is use the antibodies that we get from the rabbit. So we've injected the rabbit with SARS-CoV-2 protein, it's created an immune response, we've bled that rabbit, extracted the anti-SARS-CoV-2 antibodies from it, specifically just those antibodies, and then we can label those antibodies with some sort of coloured marker, sometimes fluorescence, sometimes an enzyme, but in this case just a very strongly coloured chemical. And that chemical is quite often what we call colloidal gold, but it can be one of lots of different things. But it's just a coloured, it's an antibody with a coloured tag on it. So this is a test strip, and it works in a very similar way to a pregnancy test kit. What we have got is an anti-SARS-CoV-2 antibody bound to the test strip here, and then we've got another second antibody bound a little bit further up. That's just the control to let us know that the whole procedure has worked. So what we've done, this is the anti-SARS-CoV-2 antibody, and this is the saliva or blood sample. Probably in this case it would be a saliva sample because we are doing an antigen test. So if you were infected with virus, you will have antigen within your saliva. So you put a small amount of saliva on the end of the test strip with some liquid just to encourage the substances to wick along this test strip. So you do that. When that analyte potentially containing the virus starts to migrate along this test strip, it will dissolve this labelled antibody which is being applied to this bit of the test strip. So as the saliva mixed with some liquid draws along here, this will detach because it's very loosely attached to this test strip, and this antibody can bind to this viral protein here. So we get, because this antibody is a anti-SARS-CoV-2 antibody, and so is this one. They both bind to the virus. This one is labelled, this one isn't. So as this analyte comes along, if there's any virus present, it will bind to this antibody, and then the whole lot will flow along this way, and if this analyte is SARS-CoV-2, it will bind to this antibody that is already bound to this antibody. So here we can see the virus protein bound to the label antibody flowing along, and then it's going to come into contact with this antibody which also recognises this viral protein, and all three stick together. The immobilised antibody that stuck to the test strip, the viral protein, and the labelled antibody that detects the viral protein. And this gives a coloured line on the test strip, very much like a pregnancy test. Now the control line here is also another analyte that is down here, and that is just to prove that the saliva sample here made it to here. So it's another labelled protein, an antibody, and we're not going into the details of that, but that's just to prove that the sample made it this far. So this will always be positive. This line will only be positive when there is virus present. So this test is an alternate to the PCR test. It will tell you whether you have got active virus within your saliva. The problem with this is the sensitivity and specificity is not as good as PCR, and we'll talk about sensitivity and specificity next week. Now the third and final test is the antibody detection test. So as we talked about earlier, if you're infected with SARS-CoV-2, you'll create antibodies to it, and those antibodies will be floating around in your blood. So this test system relies upon isolating some SARS-CoV-2 antigen, so it could be the spike protein, and what we do is we label the spike protein with a label. In this case, it's colloidal gold. So there's the COVID-19 virus protein labelled with colloidal gold, very strongly coloured. That is present on this first part of the test strip. This is where we put our sample. So we're going to take a blood sample, apply it to this bit of the test strip, and just like before, that's going to flow along the test strip. When it goes along the test strip, it will encounter labelled antigen, and it will also encounter some, in this case, chicken antibodies, and that's just part of the control to give our positive line at the top of the test to prove that the test has worked. So I'll just take you through all the bits that will rely on this test working. If you have had SARS-CoV-2, in your blood you will have one of two different types of antibody, IgG and IgM. These are two different types of antibody, both naturally present in blood. Okay, so you put your blood sample here, and that's where you put your blood sample there. This is a representation of what is inside this test strip. Okay, so your blood sample goes there. Your labelled SARS-CoV-2 protein is on this conjugate pad here that just is very loosely attached. The blood flows along there, liberates this antibody and this antibody. Now, what we're trying to do here is find out whether we have got anti-COVID-19 SARS-CoV-2 antibodies. So on this line here, we've got an antibody that recognises human IgM, and this line has got antibodies immobilised, which are human that recognise human IgG. So if you had an infection with SARS-CoV-2, you'll generate antibodies to either IgG, IgM, or possibly both. Now, this antibody will always bind, this is the IgG, will always bind to this immobilised antibody here. So that orange one is that orange one there. this yellow one is this one here, yellowish. Okay, now if you have this antibody in your blood, that antibody will bind to this protein which is labelled and then effectively deliver that labelled protein to this line here. So the anti-IgG antibody will capture the IgG, and the IgG is from your blood, which has captured the SARS-CoV-2 protein. Similarly, if you've got IgM in your blood, which is this blue one, that will capture the SARS-CoV-2 protein, and then this complex of this blue one and this one will be captured by this grey antibody, which is an anti-human IgM. The end result is, if you have antibodies to SARS-CoV-2 which are IgM, you'll get a line here. If you've got antibodies which are IgG that recognise SARS-CoV-2, you get a line here. And if you have no antibodies to anything, you'll always get a line here, because there is a labelled chicken IgM, labelled with colloidal gold, and that will be picked up by this anti-chicken IgM, which is here. So to summarise, you put your blood sample in there, the blood sample wicks up here, it will carry the labelled protein and antibodies with it. If there is no viral proteins present, your IgG and IgM cannot be captured, cannot capture any of this colloidal gold labelled protein. If you don't have antibodies to this, this will just carry on floating off up the test strip, and it will not bind here or here. Whereas if you've got IgG, the IgG that will recognise the SARS-CoV-2 protein will get captured by this antibody here. You'll get a positive line here. If you've got IgM, anti-SARS-CoV-2 IgM, you will get a positive line here, and if you've got both, all three lines will show up. So just like on a pregnancy test kit. Okay, so for discussion in the session on Wednesday, we will discuss the benefits and weaknesses of mass testing. So we're currently testing a lot of people who are negative. We're possibly getting a few false positives, which could be causing problems. We could be having tests which are not 100% accurate, not 100% sensitive. We'll be discussing the relative weaknesses and benefits of the viral PCR test, the antigen test and the antibody test. We'll be discussing what a positive result means for the patient. We'll be asking whether a positive PCR for virus always means you're infectious. Sometimes it doesn't, and we'll discuss what the implications are for that. And we'll also be discussing negative tests. Some of you may well have suspected you've had this infection but come up with a negative test. Should someone with symptoms and the negative test self-isolate? If they should, is it worth having the test? So these are the sorts of things that we can have a think about and have a discussion in the live session. Okay, that concludes the COVID-19 session 4.